The talk will be about molecular determinants of oxidation dependence of volume-regulated anion channels. I will first give some introduction on these volume-regulated anion channels. Regulatory volume decrease is a property that most cells have. Most cells can regulate their volume. So, for example, upon a hypotonic challenge, cells swell, and then there are various mechanisms, channels and transporters that lead to the loss of osmolytes from the cell, and then can get back to the normal volume because water is then leaving passively and they get the volume back. And one key player in this process is this channel here, the volume-regulated anion channel. And this is an ubiquitous channel. It is practically found in all cells of the body have it. And it had been actually investigated for many, many years, for example, by Thomas Fuchs, who has like many, many papers on this channel. And only rather recently, so this is like a typical patch-like experiment. You patch the cell, and then you apply a hypotonic solution, you get huge cell swelling, and you get with time activation of this sometimes time-dependent currents, which are carried mostly by chloride in these conditions. And for many, many years, the genes underlying this channel have not been known. And only in 2014, two groups in parallel identified the so-called leucine-rich repeat-containing 8 proteins as underlying this volume-regulated anion channel. And actually, the proteins had been described already earlier just because of sequence similarity. And Abascal and Zadoya, they found that the proteins, these LRRC8 proteins, most likely have 4 transmembrane domains, and they have a channel-like appearance, and they have some similarity to pannexins. Pannexins had already been known before. Pannexins have also the same topology. There is some sequence similarity. In addition, this is also the name-giving feature of these proteins. They have a leucine-rich repeat at their C-terminus. So there are like 18 or many leucine-rich repeats at their C-terminus. And indeed, then functionally, it was found that indeed these channels are heteromers. They are not monomers. They are multimers. And importantly, I will briefly call them 8A because it's a little bit tedious to call it LRRC8A. So if you just express the 8A subunit alone in a cell line that has no endogenous LRRC8 gene, so this is a knockout cell line, there is no current at all. You need another subunit. You need, you need, for example, to co-express the 8A subunit and the 8C subunit, or the 8A plus the 8D, or the 8A plus the 8E. Then you get nice volume-regulated currents. And none of the other subunits alone give currents also. So you need a combination of at least one 8A and at least one other subunit. And having now the genes, there have been also now various knockout studies. I don't go into the details, but the VRAC or the LRRC8 is also important for other physiological processes. For example, in beta cells, it's involved in insulin secretion. It's very important for sperm maturation, where actually the volume decrease is very important. Then in the brain, it is discussed that VRAC is important for the release of excitatory amino acids, like glutamate, for example, from astrocytes. It is important in apoptosis. It is also important in several instances in chemotherapy resistance, for example, by platinum-derived compounds uptake. And it's also, for example, important in the so-called STING pathway in immune cells and in cancer cells, where it's important in the transport of cGAMP. However, and also, it was surprising, there were like 4 studies in 2018 that reported the cryo-EM structures of the homomeric LRRC8, and they all converged on the finding that this is a hexamer. So this is the homomeric LRRC8. This is not physiological, but it at least shows that the architecture of these proteins is hexameric. They have a relatively narrow pore entrance on the outside, then have a large pore here in the transmembrane domains, a cytosolic linker region, and you have this intracellular leucine-rich repeat domains that in the homomeric protein, form these dimers. It's a trimer of dimers somehow. So there's a break of symmetry from a 6-fold symmetry to a 3-fold symmetry at the C-terminus, which is rather surprising. Despite all this knowledge, the mechanism of activation, so how hypotonicity activates this channel, is still not known. A leading hypothesis that has been put forward by the Nidius lab, and also Thomas has worked on that, is that these channels are activated by low intracellular ionic strength, even though it is not really clear how physiologically if the ionic strengths can really go down to levels which are enough, which are sufficient to activate the VRAC. Then reactive oxygen species have been implicated in the stimulation of VRAC. And then there has been also a large body of literature on the role of various phosphorylation events in the regulation of VRAC. And also recently there has been a paper by Tobias Stauber's group involving phosphorylation as one of the trigger events in VRAC activation. In my talk, I will concentrate on the regulation of these channels by oxidation. And in previous work, we had found, in collaboration with Raúl Esteves, that when we co-express the 8A and the 8E subunit, we get a dramatic activation by this oxidizing reagent chloramine T. So you get like almost more than 10-fold increase in currents, which can be blocked by the blocker carbinoxolone. And using various reagents, cysteine reactive reagents, we could conclude that this oxidation activation probably regards the oxidation of intracellular cysteines. On the other hand, quite surprisingly, the other subunit combination, 8A plus 8C, was instead inhibited by oxidation by chloramine T. And again, using these compounds, we concluded that it's mostly the inhibition of 8C is probably mostly mediated by the oxidation of methionines. And the goal of our work that I present here was what are the molecular targets of oxidation for the activation of 8A, 8E, and for the inhibition of 8A, 8C. So if we first did chimeras, we put, for example, the C terminals, so the LRR domains from 8A onto 8C, and in that case, indeed, we could restore activation. So normally 8C is inhibited. This chimera is activated by oxidation. And the reverse chimera, if we put the LRR domain from 8C onto 8E, we get indeed inhibition by chloramine T. So we can swap the response by exchanging the LRR domains of the respective subunits. Then we did a lot of chimeras. I don't go into any detail, but then after all these chimeras, we could, at least regarding the 8E sub-oxidation-mediated activation, we could restrict our search to there are 3 cysteine residues which are present in 8E but are not present in 8C, which remained the final candidates. And then here in the final countdown, if we mutate all 3 of the cysteines into non-cysteine residues, we lose the in activation by oxidation. If you mutate the 493 cysteine, we retain activation by chloramine T. If we mutate both of the other cysteines, we again lose the activation. However, somehow surprisingly, even the single cysteine mutants of these 2 completely abolish the activation by chloramine T. So both single mutants, as the 424 and the 448, lose sensitivity to chloramine T activation. From this, we conclude that both mutants are needed, so both residues are needed, and we speculate somehow that, so if you look at the structure, if you map these cysteines, there is no structure of the 8E, but if you map these residues, they are relatively close to each other in the first part actually of the LRE domains, and we speculate that oxidation could lead to the formation of a disulfide bridge, and indeed this activation is reversible by DTT, so this is some indication that there is formation of a disulfide bridge. And to test more directly if there is formation of a disulfide bridge, we applied a crosslinker. So we expressed 8A and 8E, again in cells devoid of endogenous LRRC8 subunits. Then we perfuse a small cysteine crosslinker, BMOE, into the pipette, then we get gradual increase of current. So this is a strong indication that indeed the crosslinker can activate the current in the absence of any hypotonic stimulation. So this is indeed an indication. We don't We don't see this activation if we express the double mutant. So this is gone if we express, we eliminate the cysteines. And we can now also ask the question, is this of any, does this also happen in vivo, so in normal cells, in non-transfected cells? And for this, we just perfused the same cross-linker now in non-transfected HEC cells, which are endogenously express the VRAC current, and also in this case, the BMOE activates typically VRAC currents, and they are not huge, and this tells us that at least a certain fraction of the endogenous VRAC currents can be activated directly by this disulfide formation between the two cysteines. Good, these were nice results for the 8A, 8E subunits. So we could identify the target of oxidation, but what about the 8C? And for 8C, somehow the chimeric strategy was not really conclusive. We could not draw any nice conclusion in that. Then we actually mutated all methionines, all cysteines, and even some histidines of the 8C subunit, and we could not eliminate in any means the inhibition by oxidation. However, we did not touch one methionine, which was the first methionine, which is of course difficult to mutate. If you mutate the first methionine, proteins will have difficulty to be formed. And one simple idea could be, okay, let's try, how can we test the first methionine? One idea is we could make concatamers. We can make a concatemer of 8A and 8E, or 8C, and in that one we could mutate the methionine. However, it was known that the N-terminus of the protein has to be free. If you fix it in the concatemer, it is not functional at all. So this was already known. So we had actually Paulo, who is here, he had the idea, okay, do the concatemer, but separate the two subunits with a self-cleaving peptide, T2A. In that, you have only one protein initially, but at the translation step, the things are cleaved, and you have actually two, you free the N-terminus of the 8C subunit. And so we did this, and first of all, we did the control concatemer. So we make a concatemer of 8A and 8C separated by the self-cleaving peptide, and this is inhibited by chloramine T, as is the wild type 8A, 8C. So this works, at least in the control. When we then mutate the first methionine in the second subunit here, indeed, so this is indicated by an L because we mutated the methionine to leucine, you completely abolish inhibition by oxidation. And now we can also do more to this. We can now, which is like an oxidation insensitive mutant, we can introduce the two cysteines that are present in the 8E subunit. You see what happens in this case. And indeed, when you mutate the first methionine and also add back the two cysteines of the 8E, we get actually activation of this construct. So this is actually, I'm practically finished. This is just the conclusions. So we believe that oxidation of the 8E subunit involves formation of a disulfide bridge in the 8E subunit, and being in the initial part of the LRE domains, we think that the disulfide bridge somehow constrains an opening of, induces an outward movement of the LRR domains and thereby opening the channel. And this might be, I mean, normally in intracellular conditions are highly reducing. You would not expect formation of disulfide bridges, but under certain conditions, and especially in very localized conditions, you might achieve high reactive oxygen species concentrations, for example, in immune cells. So this might actually be a physiologically relevant mechanism which remains to be tested. And we believe that the first methionine of 8C is target of oxidation and inhibits the current. And there is some evidence that the N-terminus actually goes into the pore. And we believe that, and it's forming part of the pore, and because mutations in the N-terminus also lead to alterations in ion selectivity, and we believe that oxidation of the first methionine might actually lead to a direct occlusion of the pore and thereby inhibiting the current. And last slide, I would like to acknowledge that this work was actually, most of it was done by Sara Bertelli during her PhD thesis in Genova, and Paolo Zuccolini contributed, and we had also a collaboration with Raúl Esteves from Barcelona and some funding, and thank you for your attention.
Many thanks for the talk. Hello, it's here. Pardon my ignorance in the field. Does this channel have any inherent mechanosensitivity, like people applied force and see?
No. So if you, I mean, if you push on the membrane, you don't activate the channel. There doesn't seem to be like a mechanosensitivity in that. It needs, you can activate it without hypotonicity by other chemical stimuli. For example, in some cells, you can perfuse the cell with GTP beta S, and you can, or GTP gamma S, sorry, you can activate it without hypotonicity, but just swelling the cell mechanically doesn't activate the channel, doesn't seem to activate the channel.
I've got a second very small question. There are many leucine repeat domain containing proteins which are known to mediate protein-protein interaction. So are there anything known for this channel, whether that domain is?
So there are candidates of protein-protein interactions, for example, also from the cytoskeleton, but there's no real mechanistic link so far.
Thank you.
I had two questions. The first one is, so you now have a hexamer, and there is possibility that you have more than two subunits in one, right?
Yes, yes.
So what did you try, for instance, what happens if you have the one which is activated and one which is inhibited by the oxidation in one mix, let's say, whether you get a mixed effect? Or?
We did not try to co-express more than two, because then, no, we did not do that, yeah. This could be interesting, yeah.
Another thing I remember from old experiments is that you could inhibit activation by one mechanism with the other. For instance, that if you activate it with GTP gamma S, that you could inhibit it by shrinking the cells. Do you think does this oxidation mechanism prevent any other activation mechanism, or do they become insensitive to swelling or shrinking?
They become insensitive. At least, I mean, if you strongly activate it with the oxidation, you not further activate it by hypotonic swelling. So it's dominant somehow, the oxidation-dependent activation. It depends on the degree. I mean, if you don't fully activate it, you can still further activate it by the hypotonicity. But if you strongly activate it by oxidation, then you cannot further activate it.
But could you inhibit it by shrinking? Or?
You can inhibit something by, you can also reverse partially by shrinking. So it's like, it looks like it's in the same pathway somehow.
Michael, do we know what is the diameter at the level of this cysteine in the cavity?
What is
the diameter of the
distance of the cysteines?
Yeah, at this level of the channel pore.
I mean, we don't know because we don't have the structure of this subunit. But if you map,
you try to model, let's say, to approximate the.
So here, if you look, the distance between the two residues in the 8A subunit and the structure of the 8A monomers is like 7 angstroms, which may be too far really to do. But if you force it to, I mean, they're flexible, and they may once in a while reach a distance that you can allow, that would allow a cysteine disulfide bridge.
We're all very interested in lunch. Anyhow, this was excellent. Thank you, everyone.